NCBI BLAST, that is Basic Local Alignment Search Tool, is widely used for sequence alignment. NCBI BLAST can identify similarity between biological sequences such as DNA, RNA, or protein to reveal evolutionary relationship. The next one is functional annotation. NCBI BLAST annotate unknown sequences by comparing them to a databases of characterized sequences to interfere potential function. It can also help in identifying potential drug target by searching for the sequence similarity in genome of pathogen or disease-related genes. NCBI also can help in analyzing complex microbial community by comparing DNA sequence to identify and characterize the diversity of microorganisms. And lastly, it can also be utilized in reconstructing the evolutionary relationship and constructing the phylogenetic tree based on sequence similarity to understand the evolutionary history of the organism.